The pros of shrinking the lab are that we're able to go to places that we couldn't previously go with the sequencing. So by having a small laboratory, we're able to deploy much faster in a much more cost-effective manner to more places around the world. What we're looking at here is the sequencing workspace. The main benefits to sequencing in the field, firstly, it's speed. You're able to take the sample to sequence within a very short period of time without having to take the sample out of the country at any stage. What that means is the country keeps ownership of their samples. You've got the capacity to train the local staff in how to do the sequencing, and it allows you to report the results much faster than if you had to send the samples out of the country. One of the problems we have in the field is making sure that we can power everything, and we want to be able to work in very remote locations when it's required. So to get around that, all of the units, everything that we've put into the sequencing kit is designed to run off 12 volt maximum, so that we're able to power it either off lithium-ion batteries or off solar power. You can see we carry with us 2 foot by 1 foot Goal Zero solar panel, along with this power pack called the OT150, which is sufficient to power the entire unit for 12 hours, even in the absence of sun. We use sequencing because it allows us to track the virus or the pathogen from one person to another. So by looking at the genetic level, we can say whether the differences in one virus have been transmitted to another person. So what we'd normally do is the first thing, we'd come into this chamber and we would set up the sequencing reactions. Once you've done this, you take the sample out of here and you come around to one of the PCR machines. We've got the mini PCR, which is the one we use when we want to make libraries and amplicons. If we need to quantify the DNA first, then we'll use our qPCR machine, which is called a MyGo, which can do 16 samples at a time. This is the MinION sequencing unit from Manipur. As you can see, it's very small, it's very portable, and it provides us a greater flexibility in the field. You can see it's small, it's light, it fits in our bag, and it's quite accessible for people to use. Mobile sequencing needs to be portable. 2 people need to be able to carry the sequencing facility to anywhere in the field. It needs to be robust. It needs to be able to handle harsh environments such as high humidity, high temperatures, pests, and similar things in the field. It needs to be rapid. We need to be able to go from setup to sequence within hours, not days. It needs to be fieldable. It needs to be able to cope with the absence of air conditioning, cold chain, power, or internet. It needs to be simple and trainable. We need to be able to train local staff in how to do the sequencing themselves to provide capacity for future outbreaks. It needs to be accurate and interpretable. We need to be sure that the sequences we're getting are correct and that it's easily reportable to people within the field. Finally, it needs to be cheap. We need to be able to sustainably fund this for future outbreaks. The initial cost for setting up the laboratory are around $15,000, and the cost per sample aims to be between $5 and $10 per sample. With the laboratory we've developed, we aim to sequence 100 samples without the need to restock from outside the country. The laboratory is designed to be able to operate for 4 to 6 weeks in the field in the absence of cold chain or power. Everything in the kit's designed to be accessible, and we've listed everything that goes into it on the website arctic.network. Next for the project, we want to continue trying to make things cheaper, smaller and lighter so that we can go to more locations around the world.